So today we will receive Professor Amadeu Sum. He is a professor in the Chemical and Biological Engineer Department at the Colorado School of Mines and leads the Phase to Flow Laboratory, the laboratory that I had the pleasure to visit and stay for a while. And thank you for that, Professor. He has been working with Clathrate Hydrates research for over 20 years, covering experimental, theoretical, and simulation topics of hydrates in flow assurance and science, passing by phase equilibria, kinetics, deposition, chemical inhibition or management, interfacial phenomena, multi-phase flow, spectroscopy, and molecular simulations. He is author and co-author of over 194 reviewed publications and has a book on hydrates in flow assurance he is the creator of two inventions. He has given a number of lectures and short courses on hydrate to academy and industry and has worked closely with industry, including hydrate dips and dip store. In his talk, he will present new finds and insights into the molecular thermodynamics of Clathrate hydrates that are relevant from molecular simulations. He will also discuss how simple thermodynamics are applied to solve the all predictions of hydrate thermodynamics with so-called inhibitors. We welcome you, Professor Amadeu, again. Thank you, and the word is yours.
Thank you, Ingrid. Let me share my presentation in here. Can someone give me okay? Can you see my presentation?
Yes.
Yes. Thank you. Yes, I'm very happy here to give a seminar to the Adams group. I thank Fred for inviting me and the Adams group for inviting me to be part of this distinguished list of speakers. I've have roots in Brazil, so to be speaking mostly to the audience there in Brazil, it is close to my heart too, and being able to contribute it that way. Today I want to talk about a couple different things, and I put this title in order to sort of get your curiosity here on what do we mean by simple structures, hidden structures, and simple thermodynamics. Right? And if you look at it, the slideshow that was previewing before this talk, there was an image there showing the snow, and there's this other one that I'd like to show about mines with the mascot, that's the burro. And last week it didn't look very much different from this. We had a pretty big snowstorm, and although today is over 20 degrees Celsius, so the weather here changes very fast from one day to another. And that's one of, I guess, the nice things perhaps about living here in Colorado. We get to see, in a way, the 4 seasons in a very short period of time. I want to start this talk by this quote from Einstein that I really like. And it is one that I think drives a lot on what we do, and at least the philosophy, the philosophy in my group, in terms of how we want to think about research and how we try to understand things. And he says a theory is the more impressive, the greater the simplicity of its premise, right? And I think that it is very true for a lot of things, especially how nature does things. I think we tend to make things too complicated, whereas nature tries to do things in its simplest way. And I think this is a statement here. It is very valid for thermodynamics. Thermodynamics, in a way, the more that you learn about it, the more you get to appreciate how perhaps simple thermodynamics is. Some might, of course, disagree with that statement. but is one that has essentially wide applicability to many different areas with very few simple concepts, right? So, the more we do, the more we come back to thermodynamics, and this will be emphasized in the thing and some of the topics that I'm going to discuss with you today here. Okay. Just some initial remarks and sort of setting the frame here for what we will discuss in my talk. Clathrate hydrates, it is a very rich area for science engineering. So there has been previous talks here in the Adams seminar that have talked about hydrates, and I wanted to emphasize this, how rich this area is. Clathrate hydrates are far more complex than ice structures, and we'll discuss that. We have hidden structures here. This is, in a way, the first that I'm presenting about this hidden structures. So you're getting a preview into some of the things that we are discovering. And this last point about simple thermodynamics on how we should be thinking, and I want to essentially pose a new way of considering how to deal with inhibition of clathrate hydrates. Okay, so since there's people from various backgrounds in here, and even though there were some talks about clathrate hydrates, I want to give you my introduction to it in terms of what they are, right? And we are going to be talking mostly about the gas clathrate hydrates. And so for the gas hydrates, we need temperature, pressure, a gas, and water that combines to form a crystalline structure, right? And the image there that you see of the burning ice balls, right? That's the combustion from the gas that's being released on the dissociation of the hydrates. The hydrates have a fairly large content of gas that can be trapped in the crystalline structure. And that corresponds to about 164 times if you were to dissociate one volume of a hydrate. One of the most unique properties of hydrates is that it is non-stoichiometric, meaning that it is not a one-to integer number of water to gas molecules, and that has to do with the occupancy of the different cages that are formed. Right? So hydrates are known as inclusion compounds. Now, these are the basic water cages and hydrate structures that are most common, that most people would be familiar with. And I want to emphasize that point about the most common ones, right? There is a structure 1, there's structure 2, and structure H. And the colors here help to represent on which cages are part of which structures, right? Structure 1, structure 2 are cubic, and structure H is hexagonal type of crystal structure. Okay? And so there are many different guests that can occupy the cages, and by and large, the size of the guest will determine which crystal structure will form. And here I show a selective list for the structure that will form from the single formers, right? Of course, we can have mixtures, and then the mixture, the crystal structure for the mixture is going to be mostly dependent by the largest gas that would be accommodated inside of these cages. And just to emphasize again, the gas that's trapped inside of these cages are essentially free, right? They're not bound to the water, so it is like a prison, and that's the name of clathrate, derived from the Latin, right? So as you see, there's a whole range in here of molecules, and by our understanding, these are the different types of structures that would be formed. Structure H here needs a small guest in order to occupy the small cage, along with the large guest here that would be in the red cages that are the large ones. Right? So what is important also to realize is where hydrates are going to form, right? And that has to do with the availability and the contact of water and gas. Right? And so if we look at the solubility of water in gas, there's very little chance that the hydrates would form in a gas phase. The solubility of water in oil or that would be saturated with the gas, right? It is also a very unlikely scenario, place for hydrates to form. And then you have in bulk, these are the ratios in the bulk phase, right? You have only about 2 to 1,000 gas molecules in water. So it's an interface, right? The interface where you have the largest concentration of the different components that are required. But most hydrates form, right? We have this ratio of about 1 gas to 6 water molecules. And so this, in a way, defies or conventional wisdom that water and gas don't mix, but they do in hydrates, right? And that's why we say that we have a solid solution, right? This ratio here, it is very much what you would find in any other kind of miscible system, right? So it is, but instead of being a fluid phase, in a solid phase, right? So let me give you my view of the world of gas hydrates, right? And we have the good of hydrates, and this has to do with the potential energy gas that's stored in hydrates. There's a very significant amount, and there is interest in harvesting that gas from hydrates. As I showed you, there's a significant amount of gas that can be trapped in the hydrates for energy storage, and there is interest as well of looking at hydrates for desalination purposes or even for hydrogen storage. We have the bad side of the hydrates that's related to how it impacts mostly oil and gas production. It's a flow assurance issue, area that one of the areas that we work very intensively, and that's a solid precipitation problem. So that's what I say, it is the bad. We have the ugly side of hydrate that has to do with the problems that it actually cause in the flow lines and creating these plugs. And so one would have to remediate and remove those as they pose safety hazards, right? And there is also the side of hydrates into climate change, right? On all this gas that is trapped in the hydrates, if somehow the hydrates do dissociate, it can be a significant amount of carbon going to the atmosphere. And actually, just this past weekend, I read an article saying that there has been some evidence of this happening in Siberia with the melting of the permafrost. And so this scenario is actually kind of happening before our eyes. And so we need to be very careful here on the consideration on the climate change impact of hydrates. And then I say there's the beautiful side of gas hydrates, and that's the science, right? Hydrates are mostly water, and who doesn't find water interesting, right? And so there's a whole side of hydrates there, and that the first part of my talk has to do with this beautiful side on the things that water does and hydrate in the form of hydrates. It is even more interesting from my point of view than just water, because it involves interaction with a gas molecule, right? So whether you look at through experimental method or simulation methods, there are a lot of beautiful things to be learned there. So the research that we do, it is very vast. There's the depth and breadth in here into many different areas on the fundamental side, and then also on more the applied side. We do the experiment, the modeling, the simulation, and there would be a lot of different things that I could talk to you about today, but I was very selective because of the audience in here in terms of, I think, what you would get the most. Maybe Fred might have to invite me to some other seminar for me to talk on some of the other areas. But what I'm going to do mostly, it is on the fundamental side in here today. Okay. So one of the big questions that still exists, right, and not just for hydrates, but for any phase transition, right? It is how does that phase transition happen? This work was propelled by what my group did over 10 years ago now, right? It doesn't seem that long, but it was when we first published this paper in Science that looked at it. The first reported spontaneous nucleation of hydrates with methane being the gas, right? And so this is the video that we generated as part of that publication. And you see, in a way, the dancing of the water molecules with the gas molecules and forming the cages, and that creates a solid phase that resembles the hydrate, right? And then in this second video in here that unfortunately is not playing, you see the growth of the solid phase with the different types of cages that are being connected to form something that it is not necessarily crystalline, but it has features that resemble hydrates. So since this paper that we had published, again, over 10 years ago, there has been a significant interest in using simulations in order to further probe the molecular interactions, the mechanisms of hydrate nucleation and growth. Just roughly, there is about 30, 40 citations of this paper per year since. And so, if you add those up in over 10, 12 years, there is over close to 400 papers that have been published related to this topic, right? Or at least have cited or initial study. And I think that's a fairly significant amount of interest in this particular area, and people continue to do research using simulations to understand hydrate nucleation because the questions are now resolved, right? We have gained some significant knowledge, but there's still a lot that we need to learn. So in general, these are some broad statements in here of what one can say on what we have learned to this point. Some people might disagree with some of these statements, and I might, of course, have missed some of the advancements. But in general, we can say that nucleation and growth of a clathrate-like phase, right? That's amorphous. I don't think there has been any kind of studies that have shown a long-range order that corresponds to a crystalline structure as we know that I showed you, like structure 1, structure 2, or even structure H. We know there's numerous cages formed, much larger than the list that I showed you in a previous slide. The critical nucleus is not well defined by geometry, by shape, or the types of cages, but is defined by size. There has been many different other parameters that have been developed and used in order to monitor or track the evolution of the system. We have seen from these studies that the amorphous to crystalline annealing, it is really not achievable within the simulation time that most of us can afford to do. And I think all the studies done so far have discarded this labile cluster model hypothesis that have been in the literature before simulations really came into play. And so I think that was a significant contribution because the previous understanding based on this labile cluster were really just hypotheses. There were no evidences for it. Okay? And so when we consider hydrate nucleation, we are interested in a mechanism, right? And when we talk about a mechanism, that has to do with the size of the critical nucleus, right? And then you want to be able to calculate or determine what is that free energy barrier for the nucleation. And so we have done one of these studies and performed many thousands of simulations in order to arrive at the results that you see shown in this slide. Others have done this with slightly variation with different methodologies more recently. And so I think there has been some significant progress in here into not just the methodology, but in our understanding of the hydrate nucleation mechanism. But I think most of the studies have been consistent, which is also good. And what we see is that the critical nucleus, again, as I mentioned, is not necessarily unique. There's various arrangements of water and gas molecules that one can find, and is defined by size, not necessarily, again, by the arrangement of the molecules. And I should emphasize also that this has been done mostly for methane hydrates, right? Because that's the simplest system, and also one that has, of course, great importance, as methane is the main component of natural gas and also of the hydrate deposits that are found around the world. And so from doing such process, you can extract a mechanism, right, that would give you that path that on the ordering of the interactions of the molecules until it reaches a critical nucleus. And then from that point on, it is the growth stage. Right? Now, nucleation is a stochastic process, right? And so we need to be aware of what is the nucleation rate, and certainly a rate associated with the phase transition. It is something that one is also interested in doing. Now, as I said, hydrate structure is mainly defined by size of the gases. Okay? And I think anyone who works on hydrates would agree with that statement. But of course, there is exceptions, and I want to point out here in the next portion of my talk the exceptions. Okay? This is based a lot on what we have done in terms of studying the simulation for hydrates formed from propane and mixtures of methane and propane. Right? These are just two graphics here to represent a couple of the things that we have done. For the propane system, we have carried out simulations up to 20 microseconds in here to see the annealing, but nothing really happens in that, even in that time frame that it is long for simulation, but of course in real time it's not long. And then for the methane propane here, we have seen this diversity of structures that are formed, right? And so let me touch upon on some of this. The simulations that we have performed were for different compositions of ratios of methane to propane, right? And these are just some of the summary of the conditions and the number of simulations that we perform. And altogether here, you can see that we have generated over 300 microseconds of data. And one important point that I want to make is that for us to do these studies, we do many simulations, not just one or two, right? You see that there in total, we had done 30 for the pure propane, 15 for the 20-80 ratio, and then also 20 for the 89-11. And that's important. Most of the studies in the literature that you see that are reported are based on the simulations of 1-2 trajectories. And if you really are trying to do nucleation studies, it is a statistical process, right? So you really don't get much information, or you don't get the statistics required for you to truly understand the nucleation. So that's why it's important that you consider as many simulations as you are able to afford to run, right? And so this is an example of the potential energy that we obtain for this one concentration of this methane propane system. And you see there's a distribution in here on the times that nucleation happens and then the subsequent growth. And that's important because, again, nucleation is a stochastic process, right? You don't want all of them to nucleate immediately. And in a way, you have to do a fine tuning of the temperature and pressure so that you have this distribution of times, right? And so it does represent a spontaneous stochastic process. And of course, if we were to change the conditions and not have any nucleation within the time that we can simulate it, that doesn't mean that the system, the conditions are incorrect. It's just that the time that you are looking at it, it's not sufficient for these events to occur, right? And so that's another important point to keep in mind. And because each trajectory here has very important information, we want to make sure that we consider the information that we have from all trajectories, not individual ones. Each individual one, it is not necessarily that relevant in the scheme of, again, the stochastic nature and the probabilistic nature of nucleation. So we really want to use all the data from all the trajectories to understand the nucleation process. One important thing also that I want to emphasize is how you analyze your simulation trajectories. We have all the data, but you are only going to learn as much as you try to extract from it, right? And that's part of what needs to be developed. And for this particular study, we develop our cage cluster recognition code in order to extract faces that can be put together to make cups. And the cups can be then combined to form cages. And then the cages can be looked at it on how they are connected to each other to form the larger clusters, right? And so we have developed this particular code in order to do this. This is nothing more looking at connectivity in terms of distances, and then adjacency matrices in order to find what is connected to what. Right? And through this process, we are able to identify then a number of different cages that are most important when we look at the nucleation. And these are cages that might be empty or occupied, and that's another important factor to consider, and something that has not necessarily been given a lot of attention in the literature. Besides identification of the cage, the occupancy is important. Okay. And so from this set of simulations, we are able to extract a critical nuclear size, right? And we are looking for the range of the critical nuclear size where we have a probability of either growing or shrinking, right? And depending on the time that you consider for this event to happen. And so we are able to identify this for the different systems. And you see here this critical nuclear size in that range of 7 to 9, 8, 9, depending on the systems. And this is again for mixtures that we are talking about. And at the bottom, you have some examples of these critical nuclear sizes at that point of 0.5 probability. Okay. And then for this particular systems, we were interested in see what actually forms after it nucleates. And so these are some images that we obtained that show that in this particular case, as with the combination of methane and propane, we actually start to see a lot more of crystalline type of structure. And this is shown here from the top row in blue, that you see a lot of things a lot like structure 1. And in the bottom row here is a lot more like structure 2. And the one that's labeled G is sort of a combination of structure 1 and structure 2. So unlike in previous studies with just pure methane that it was mostly amorphous, having methane propane, we actually induce a lot more of this order, long-range order and crystallinity. And so we can quantify that. And one way is by looking at the number of connected cages, especially in structure 1, we have rows of this 51262 cages, the green ones that are shown in this image. And we see that the degree of crystallinity here, it doesn't get very high. But again, we have a fairly, if you will, a small simulation box. And then you can do something similar for looking for structure 2 patterns, right? And the orange cages here are the large cages. And so if you look at the arrangement of those orange cages, they are in a tetrahedral orientation. And so you can track that, and you can see that it is much higher in this particular case, as expected, right? Since propane will induce the formation of structure 2. Now, let me play a couple videos in here that illustrate this. It's always, I think, interesting to look at the videos from the simulations because we get to learn a lot, right? And you can see there the formation of the structure 1 patterns. And this is an illustration. If it plays, I'm not sure. I'm going to have to play manually here for structure 2. There. And so these are formerly fairly large clusters, and which is, again, unlike many other cases that have been reported previously, where mostly was a amorphous structure. Right? Now, by looking more closely to this simulations, what kind of cages were formed, we see something like this, right? And what is interesting about this is that we really had to spend some time, do the manual work, and using the software that we have for visualization to target specific structures and try to understand what we were forming, right? And this was the hard part. And I've always asked my students to spend a lot of time to do this, because this is how we are going to really learn what we need to look for, right? And that's generally true, right? If you don't know what you're looking for, you're never going to find it. But it requires some effort in order to try to identify what does this structure that results from the simulations correspond to. Now, what I have boxed in red there, they're actually the structure one, right? And we have two structure one domains in here that are showing this yellow and the blue cages, but you see that they are not aligned. They should be aligned. Now, when we look this more carefully, what this corresponds to, this larger structure that you see here, it is actually another structure that's not as well known for hydrates, right? And there's a very large domain of it that we find in the simulations. And this is actually a well-known hydrate structure, not as common, that is known as the TS, structure one. And there's a corresponding similar structure that is known as the HS for the hexagonal structure one. Now, if we had not spent the time to really try to look at the arrangement of those cages, we would have never found this. Now, it is arising because of this misalignment of 2 unit cells of structure one that generate this 5, 12, 6, 3 cages. And they arrive because of they are trying to release the geometric frustration associated with the misalignment of structure one crystals, right? And so we can see that these two particular structures, the hexagonal and the tetragonal, they have a common building block that is shown in here. And it's just they differ by how these building blocks are put together, right? And you can see that as they're illustrated with this diamonds, right? And what is, and this just emphasizes that pattern without showing actually the cages. And what is interesting is that this particular pattern also arises at the interface between structure one and structure two domains, right? And so what we have found as these hidden structures are what it is. This is tetragonal structure, TS1. And because we've seen in here, we think that this is actually much more common than most of us think. And it is maybe actually formed in any condition that you might have structure one and a combination of structure one and structure two. And I think to this date, it hasn't been given enough attention because most people haven't looked for it. And so I think it is important that now, whenever we are trying to either weather simulations or experiments to search for this the TS structure. And that's what I mean by the hidden structure from my presentation title. Now, when we look at this simulation that we performed in 2009, this TS structure is actually here. It's given by this green cages. And again, back then, we never looked for it, but it was there. And I think this is true for essentially every single simulation study has been published to date. No one has looked for this particular pattern corresponding to the STS structure, but it has been there all along. So this is a big lesson, I think, especially for us doing fundamental research. We really need to dig deep into it to try to discover something new, right? Well, it's not necessarily new, but we just found something that was hidden. Right? Let me skip this for the sake of time in here, since I'm getting very close to the end. Let me just point out a few challenges here for hydrate nucleation. I think what requires of us doing that's really going to advance our understanding of the molecular processes for this phase transition, we need to consider milder conditions, a higher temperature and lower pressure. We have a very significant driving force on the simulations that are done, and that's because the computational resources, right? We want to have nucleation within a certain time range that we can afford to simulate. But that's going to be, I think, a major challenge going in. And I think the processes that we will see is going to be quite different. It's going to depend a lot on the driving force. Consider much larger systems, greater than, say, 10 nanometers in size. So we have opportunity to actually form this crystalline long-range order of alignment of the cages. Mixtures like we've done, but perhaps with other systems and more complex mixtures. I think it is very important that we move away from NPT type of simulations because they are not necessarily physical in the sense that the temperature, the energy change associated with the phase transition is very significant, but we have instantaneous adjustment of the temperature. So this can be done perhaps with the NVE, but also in the composition of the system. As we deplete the molecules from the free phase, then we are changing the driving force for the formation, and that needs to be taken into account. And just like I believe I demonstrated to you, we do need better analysis tools in order to extract more information from these trajectories. I'm going to try to do the next part here very quickly in 5 minutes in here, so we have time for questions on the simple thermodynamics. And I think I can do this relatively short because it is simple. What I want to think about here is how we are considering this contribution of the water activity into the hydrate phase equilibrium, right? If you write the van der Waals and Plateau model, you have this expression for the chemical potential of water, right? And you have this contribution due to the water activity in order to account for what might be dissolved in water, right? Especially when you have inhibitors. So this, we all know, and we all have used very successfully, and it works really well. But when we consider inhibited systems, we are adjusting for the contribution of the inhibitors, that is whether you're talking about organic or inorganic inhibitors. For simple systems, it works okay, depending on the models that you use for, again, the water activity. But it's crucial that you have good data in order to do that, because you need to adjust the parameters here of the the activity models to match data, right? And so let me skip this. Instead of doing that, why don't we think about how inhibitors impact the freezing of water? Because hydrates are essentially the same as water. And so we can look at this displacement of the curve based on what happens on the colligative properties, right? This is classical thermodynamics, and this is what we do to account for the freezing point depression of water. And hydrates from this point of view are essentially the same as ice, right? The solute, whether we're talking about organic or inorganic inhibitor, are not part of the solid phase, right? They remain solution. So they're only going to impact the water activity. And so I'm giving you here the two ways of expressing this suppression or depression for water and for hydrates, right? They have the exact same form. And the water activity here is exactly the same, right? And so if we consider this delta T, right, that it is relative to the fresh water, right, we can actually obtain the hydrate phase equilibrium by simply knowing what is the suppression temperature, freezing point depression. So you don't need to make any measurements of anything else. If you know what is the freezing point depression, you can determine what that is for the hydrate, right? And we have done this. This has been some blind samples that we had a known composition. We measured the freezing point depression just for ice, right, for water. And then we use that information to calculate what is this boundaries in here and compare it to actual data. So this works. So this is part of what I say simple thermodynamics, okay? And you don't need to make this very difficult and expensive experiments at high pressure, high temperature, and to make several different points in order to trace this boundary. You can do this by making one single measurement that's very simple, and typically you would do with a DSC at atmospheric condition, and just determining what is this freezing point depression for water, okay, to generate this whole curve. Now, the other thing that is important we are aware, it is that this data that I showed you here, for example, needs to be thermodynamically consistent. And we have developed 3 criteria that need to be satisfied in order to check for the thermodynamic consistency in terms of the linearity of the data, the constant value of the heat of dissociation between the uninhibited and inhibited system. And from what I just told you about the depression temperature, that's related to the water activity. And we know that the water activity is relatively constant over a relatively small temperature range. And so we need to ensure that that's true. But believe it or not, most of the models that are used out there do not follow this trend, right? And so that's something that needs to be checked. Now, we have developed a correlation that we call the Hooley-Sum correlation that captures this water activity for any salt, any mixture of salts. And the key parameter here that we have introduced is what we call the effective mole fraction. If you are familiar with electrolytes, typically one uses either molality or ionic strength. But if we plot the same data, either with molality or ionic strength, we won't collapse this data. And this data are for many different salts. But using this new concentration variable, we do. Now, so this has certainly much larger implications beyond just what we're doing for hydrates, also in electrolyte science, right? But the key point here is that we can collapse this data and have a very simple correlation for the water activity to this effective mole fraction. And we can do this not only for the electrolytes, but for the organic inhibitors. And using this, we can get a very nice simple correlation in order to calculate the suppression temperature based just on the contributions of the salt in the water activity or the organic inhibitor in the water activity. All right. And so let me skip this for the sake of time. And so I want to introduce a new paradigm here on how we deal with hydrate phase equilibria for inhibited system, right? We know that the van der Waals and Plateau model, it is accurate for an inhibited system, fresh water, and so we can use that. We can estimate that beta parameter that we require in this formulation. And then we can get the water activity very easily from either our correlation or by making a single measurement of the freezing point depression. And then we can calculate what is that suppression temperature. Now, the limitations here is for weak electrolytes. We are working on that. And also for acid sour systems, because that has also a contribution into the water activity. And we're also working on that. Right? And so, in summary, I think we have developed a very simple way of looking at the hydrate thermodynamics in here for hydrates that doesn't involve anything really complicated with very fancy activity models in order to determine how they impact the chemical potential of water in the hydrate and the fluid phase. So I'm up on time in here, so let me just finish up with what I started. Hopefully I have shown you that the science and engineering of gas hydrates, it is very rich. The hydrates are far more complex than ice structures. There are hidden structures that I think deserve a significant amount of attention now as we look more closely into simulation and experimental data. And I know that I went fast, but I hope you got a flavor for the simple thermodynamics that we can use for hydrates. With that, just want to acknowledge my collaborators, the sponsors, the people involved with some of the work that I have presented to you. Okay. Thank you, everyone, for participating, and I'll be happy to take any questions if there are any.
Thank you, Professor. Really nice presentation. I really like the slides that you present, the good, the bad, the ugly, and the beautiful of the hydrates. Really nice. So we are now open for questions. If anyone has a question, you can write down in the chat or turn on your microphone and ask. Feel free, please.
Can I start?
Sure.
Thanks, Amadeu. Very nice pictures and very clear presentation. So I enjoy a lot.
Thank you.
I wonder about the fluctuations, the sulfur nucleation, and how large are the fluctuations. And can you put these pictures I wonder how you treated these data. You have several data. Oh yeah, this one. That's very interesting picture. So I wonder how you treat these different trajectories. It looks to me that you need some special treatment about these, and also I don't know if you are familiar with can-do work that define caliber to study trajectories, kind of statistics of trajectories. Have you ever seen this kind of approach?
Uh, just a little, not very much. I maybe need to dig deeper into those. But certainly, if you take any one trajectory, right, there is fluctuations, and that's normal, right? Just because of the dynamics of the system. But we essentially combine the data for all the trajectories in order to extract useful information. We don't treat each individual trajectory on its own, right? So one of the properties or features that we are trying to understand here was, for example, what happens before nucleation, right? Meaning before the potential energy here starts to decrease, right? And so we combine the data for all the trajectories that had this period of what we call induction time, right?
Yeah.
And then we average out all that. Right? So we are really taking advantage of the statistics in here. Is that your question?
Yeah. I'm still thinking about. It's not clear to me how you can treat this exactly.
Because in a way, right, they are all independent trajectories.
Yeah.
But each trajectory, in a way, it has each trajectory has the same amount of information, right? So there's no reason to discriminate one trajectory from the other.
Well, depending on the restriction. Well, you may have some trajectory that has some more probability to appear, depend on the restriction. Like as can you put out, you have a kind of, you can define a channel entropy for the trajectories, and depending on the restriction, you may have some trajectories that are favored than compared to the other. I don't know. I'm just thinking about.
Mm-hmm. Yes. We start all of them with the same initial state from the point of view of configuration, right? We randomize the velocities, right? So they're all at the same initial state in terms of position. Right? So they have equal probability of nucleating.
Very, very interesting.
Right? So that's why.
I enjoy a lot. I'm going to have you already published this, yeah?
This particular one is about to come out.
Okay, so I'm going to read this. One other question is about related with this nucleation study. Is you point out that you control the how high is, well, you did simulation with not so cold temperature and not so high pressure. It means you control anyway the driving force.
Yes.
We see simulation that well exaggerate in driving force, and maybe the trajectories that are not maybe physical or something like that. That's something that's very interesting that you point out. But the problem is the time for the simulation is may very high. So Firuzabadi have shown last week some simulation that he put a very large amount of methane in the water. It means if you're going to calculate the pressure for that, it means very, very, very high pressure.
Right.
Maybe not physical.
That's correct.
But I wonder how these very large driving force can change the trajectories, and they may be not understand well the reasonable trajectories for the nucleation.
Sure.
I don't know.
Yeah, no, it makes a big difference in here, right? And what I show you here is actually the final states that we consider to do this multi-simulations. We went through a process to identify this temperature and pressure, right? So that we would have something like this, because like in Abbas' work, he wanted to have the nucleation almost very quickly. And so the system was under extreme, maybe not even metastable. It was beyond the spinodal, right? And so the phase transition happened almost immediately. And that, of course, I don't think it is the conditions that we want to consider for nucleation. But the driving force is going to affect, right, the rate that nucleation will happen.
Yeah.
And so, as I was saying, it's a matter of how long can you afford to do your simulations, right? We could have lowered the pressure here, but we would have to run much longer simulations in order to get this distribution, right? And so I think that's the challenge that I think is ahead of us, because I think the nucleation mechanism will change. How the molecules come together to form the cages is not going to be so sudden like we see in these trajectories, right? It might be much more gradual if under a milder condition.
When you define the pressure, you relate this with the gas solubility, or you not relate this? Because what I missed in Abbas' discussion was if you put a lot of methane, the pressure maybe is not physical, because like 10% of methane needs a pressure that maybe is not reasonable.
Right.
So maybe the pressure should be related with the solubility of.
It is. Yes. And we do look at that. And there is a certain range that it has to be in order for the nucleation to occur, right? And in these simulations here, actually, we have the gas as a bubble, a nanobubble, right? So there's additional pressure related to that interface that you need to account for. But that certainly is a very important consideration in terms of the pressure of the system, because it will affect the solubility of the gas.
Okay, I have more questions, but I'd like people ask as well. Sorry.
I have a small question, a quick question before leaving. Thank you, Amadeu. It's a very nice presentation.
Thank you.
The question is how the statistics that you are collecting for the nucleation from the MD simulation are affected by the model that you use for the water molecule. How large are the difference of the statistics that you expect if you change this model for another water model?
If we change the water model, I think others have looked at that. We have not. And overall, there's not necessarily a significant difference from what is observed. There might be, of course, difference in the fine details into the time or the rate that the nucleation might happen. But overall, I think it is to observe the same patterns, the same type of structures that are clusters that are formed. So that's certainly something to consider, right? By and large, I think everyone that does simulation have used this tip for PIs. In terms of the size of the system, the actual size of the simulation box, that's, again, we are limited by what we can afford to simulate and the given time that we have. The bigger, the better, right? And we haven't not done any studies to look at the impact of the system size on the simulation process. So something that needs to be considered for sure.
Okay, thanks.
Can I ask a question?
Yes.
Sure.
Yes, Rajagopal. Many years ago, we worked on inhibition of hydrates in pipelines, natural gas pipelines. So I have two questions. One about your simulation. It's very beautiful simulations with beautiful presentation. You simulated along the reaction coordinate, the nucleation kinetics, right? Growth rate, nucleation. From the inflection point, I presume, from the minimum energy.
Yes.
Can you correlate this with any macroscopic kinetics of growth of gas hydrates? For example, there's an old theory of based on Flory. If you do this reaction coordinate from the minimum energy, from inflection point, it will be first order approximately.
Sure.
So are you restricted to this first order? Is it really a first order in macroscopic kinetics?
So I think the disconnect there is, again, the temperature and pressure or whatever conditions that are simulated. In principle, yes, this would be doable, right? But I think we need to map out what are the corresponding set of conditions that we have from macroscopic knowledge to the microscopic. So the 250K in 1,000 bar in simulations is not what you know as 250K in 1,000 bar in real conditions, because we have molecular models, right, that are approximate for water and methane and propane in here. But yes, in principle, we would be able to do that, but it will require a lot of work, background work to map that out.
But my question is more important. What I don't understand is, from my understanding, if you simulate from an inflection point, the type of simulation you're doing, a nucleation or growth rate, if possible, it'd be essentially first order in macroscopically.
Right.
See, but the real order parameters seems to depend on, especially in the inhibited systems, not nearly first order. The old measurements in Pittsburgh, for example, a growth rate of hydrates are not first order kinetics. This is what I was wondering. How do you correlate the molecular simulation to macroscopic kinetics?
I think it will require a lot more work in here again, because as you see, this happens very fast, right? And take this one curve here that you see my cursor. The time that it goes from the fluid phase to the growth, essentially that stops, it happens over 500 nanoseconds. Sorry. All right, about. That's nothing, right? That's less than a blink of an eye. And so this is happening this fast because the limitations that we have on what we do in simulations in system size and the conditions, right? And to I think, address what you were asking in terms of rate, we would have to have much larger systems under more realistic conditions.
The other question is about the beautiful thing is comparing different inhibitors. You showed their activity coefficients. And then I noticed that the ethanol seems to have the lowest effect. You have ethanol, methanol, salts, inhibitors. Another curve.
This?
No, yeah, this one. The ethanol.
Yeah.
At the time we worked many years ago, we tried to substitute from methanol, which is very expensive here, to ethanol in pipeline. Here, of course, there are difficulties of using glycols at the time. We thought of glycols at the time, but we use ethanol basically. What we found at that time was basically the specific heat variation with temperature is the main controlling factor of the inhibitor. In your case, it seems to, I don't know about the salt part. Is it still true, or I am out of this touch in this area quite some time? Is still ethanol can be used, or it's methanol is the only industry standard? Do they use other things for inhibition, like salts, for example?
Well, they don't purposely add salt, but a lot of fields now are producing with produce water that are high salinity, right? And so that has its own natural hydrating inhibition.
Okay, in offshore.
It is not, yes, offshore, especially in Brazil, right? The fields in pre-salt have significant amount of salt. We were talking about greater than 10 weight percent, right, salt contents in water, and so that was part of why we did this study and we developed this correlation. But brines are not typically injected as a inhibition.
How about the pipelines, natural gas pipelines? There was a problem in bends and like that, precipitating hydrates and around the curves where the pressure drop. In pipelines, where there is no brine, there is no.
Right. For gas, typically it is either ethanol or methanol. Yes, that's added. Yeah.
In your curve, apparently methanol seems to have higher interactivity at higher mole fractions, but lower mole fractions it shows almost equal activity.
Yes, right? This is activity.
Factor is the latent heat part, which you didn't comment. The enthalpy change. Which inhibitor do you think is better in terms of that? Like a variation of Cp with T, for example, ethanol and methanol. Their variation of Cp with T is very different.
I wouldn't know how to answer that at the top of my head here without going through some data. I particularly haven't looked at that. So I would have to defer this question to you.
Okay. Thank you very much. I just want to comment, many years ago, my master's student in the area went to Colorado, but he changed later on to Texas, Austin. I said, he went to Colorado, he stayed there 8 months, maybe the weather didn't suit him.
Yeah, maybe. Thank you for the questions.
Thank you.
Can I ask a question?
Sure.
Sure.
Hi, Amadeu.
Hi, Baman.
Hi, Baba.
Hello.
Still here.
Yeah, I'm still here. Very nice presentation.
Thank you.
Enjoyed the presentation. I was very interested in your work on the measuring activity. I was thinking that this probably is not suitable if your inhibitor is water soluble, or you have gases which are highly water soluble, especially high pressure, because we approach this from a different angle. We wanted to find an easy way to measure hydrate inhibition of water without actually knowing that what's in the water. So we developed a correlation between water freezing point and hydrate inhibition. But then we noticed that high pressure, they will diverge a little bit from that relationship.
Yes.
And I was thinking that, have you experienced something like that?
For the cases we have considered, no, but we haven't looked at many cases where there is this contribution of the gas into the water. I agree with you, and I think you make a very valid point there, that a high pressure, right, this activity here is going to be very different when you consider this solubility of, for example, CO2 or H2S in water, right? For the other gases, the simple hydrocarbons, I don't think this contribution is going to be significant enough to impact the total activity. Right. And part of this is accounted for, like in this, because we are doing a depression, right, from the fresh water. So if you get the fresh water correct, you should be okay when you look at the displaced curve due to the depression from the inhibitors.
Yeah. Yeah. Okay. Thank you very much.
Thank you.
Okay, do we have one more question? So I think it's time to wrap it up, right?
That's okay. Thank you, everyone. I enjoy giving this seminar, and if anyone has any other questions later, feel free to contact me.
Thank you again, Professor Amadeu.